want to thank everybody for joining us today. Appreciate our speaker today is going to be Calvin Goforth of VIC Technology Venture Development. And we have 3 speakers that will come after Calvin. So you'll be glad to put your questions in the chat box, and we'll be glad to answer those as we go forward. Before we jump into it, just want to let you know we do have a fair number of physicians that are joining us in this session, and we are offering something called physician advisory shares. Many of our companies will actually, if you're a physician and you're investing, you can also get advisory shares for doing additional work on top of that. That varies in scale and scope, but it's certainly a discussion we like to have with you guys. So in the presentations by the companies, if you want to register some interest there, we'll be glad to follow up with you through that mechanism to give you more information about that afterwards. But with that, let's go ahead and bring on our speaker here is Calvin Goforth. Calvin, can you go ahead and step up and launch your slides and start by introducing yourself and VIC Technology? Ken.
Okay, thanks. I appreciate the opportunity to be here. As Hal mentioned, my name is Calvin Goforth. For a little bit of a background on me, I formed my first company way back in 1994, and I've been in the startup world ever since, and in a variety of roles, including founder, executive, board member, and investor roles. And so along the way, I've raised tens of millions of dollars in capital, and I've participated in some solid exit events. Today, I'm going to provide my perspective on the various approaches available to individual investors for investing in high impact and high upside life science companies. So first of all, what is VIC Tech? VIC Tech is a venture studio. That's kind of the new nomenclature for what we do. When we first started VIC Tech, that nomenclature wasn't there, but basically we're in the business of forming and building companies based on the most promising life science innovation. So we're not working with outside companies. We're forming companies ourselves and building them from scratch. We have portfolio companies and operations across the US. We operate pretty broadly within the life science space, including medical devices, therapeutics, and diagnostics. And every company we form is based on a high impact technology with large commercial potential. So why did we, when we first started out, we were technology agnostic, but why did we decide to focus on life science? Well, life science innovation is just moving at an incredible pace. There's all these underlying tools from gene editing, rapid sequencing, artificial intelligence, nanotechnologies for drug delivery. I mean, there's a huge number of underlying tools that have really increased the pace of innovation. And of course, with change comes opportunity, and the market sizes are large, often in the $1 billion plus range. Now, of course, life science ventures are complex, and they have high capital requirements and significant regulatory hurdles. Often they have crowded intellectual property landscapes and more. But while that may sound like a disadvantage for being in life science space, these challenges actually limit competition at the early stage. So by putting together a nationwide ecosystem that addresses these challenges, VIC has found itself in strong position to capture some of the highest potential life science innovations and build companies to commercialize those innovations. So now the main topic of the presentation: what are some of the ways that individual investors can participate while recognizing some of those challenges of investing in the space? I'm going to describe what I see as some of the pros and cons of the 5 investment approaches I list here on this slide. So of the 5, VIC Tech is a venture studio, and we also have an affiliated VIC Investor Network, which is an angel network. So we're directly active in two of these areas. Also, before we became a venture studio, we were originally formed as a consulting firm and then an incubator. So we have experience as an incubator. I've also served on the board of directors of one of the very first equity crowdfunding platforms, and we have significant investments from various micro VC funds. So I've got experience across all these different ways of investing that are potentially accessible for individual investors. So let's start with equity crowdfunding platforms. So these are places you can go where you can review companies of all types, posting information about their investment opportunity. Well-known examples would include places like Start Engine, VIC Founder, and EquityNet, which was actually the very first equity crowdfunding platform. And that's the one that I was formerly on the board of directors for. Some of the pros of crowdfunding platforms, I mean, probably the biggest one is that there's usually a low minimum investment requirement. So sometimes it's down as little as hundreds of dollars. So you can spread your investments across a lot of companies. And there are typically many, many companies on these sites to choose from. If you have time to conduct your own due diligence, which is a substantial burden. And that's, also one of the cons. First of all, many of these companies have valuations that don't fully reflect the risks and have other poor terms for investors. And that due diligence challenge for the individual investor to sort through this large number of companies is a very significant disadvantage to participating through these equity crowdfunding platforms. There's also a pretty low due diligence standard typically by the entity. These crowdfunding sites are not putting their own money at risk. They're getting fees and percents of raise. So their incentives aren't necessarily directly aligned with yours. And there's typically no ongoing support for invested companies and no ongoing protection for the investors in the companies. So you're all familiar with angel networks. These networks range from networks are to invest in companies at a city level to international scope, and they screen companies and have those companies present on a regular basis. Some review opportunities within a specific domain like IT and tech, and some review startups in any industry. So some of the pros and cons, well, first of all, the pros, now you have other investors that you can share some of the due diligence efforts with. So that's an advantage over a typical crowdfunding site. There's, again, typically a pretty large number of investment opportunities. If you're interested in a regional focus to help with economic vitality in a given region, you can choose a regional investor network. And there's opportunity to syndicate with other angel groups. So those are some fairly important advantages of an angel network. There's some cons too. Oftentimes, and of course it varies. It depends which. I'm doing the broad brush strokes here, but it varies a lot. But many angel groups struggle with getting the resources together to conduct the needed level of due diligence. And that's especially true for complicated opportunities like life science deals. There can oftentimes in angel networks you see presenting, there's inconsistent deal quality and terms. I've gone to lots of angel investor meetings, and sometimes I really scratch my head about how did that one make it through this screening committee. But again, that varies by network. If you are focused on regional economic development, that can limit the quality of the deal flow. And typically, not so much if you've got a syndicated group with enough resources to participate in the follow-on rounds and perhaps have earned a board seat, but typically there's little ongoing support for the invested companies and little ongoing protection for the investors in the companies. So VC funds are becoming more accessible to the individual investor, and especially alumni funds and micro VC funds, where the median micro fund size is about 10 million, are potentially accessible to the individual investor. And there were a record number of micro funds formed in 2021. And from the data I've seen overall, while that data is pretty limited, the micro funds on average are doing pretty well in terms of providing good returns, or at least good unrealized returns at this point. So there's some pros. They may have professional due diligence. There's always tons of companies seeking money, so they have a large pool of applicant companies to choose from. Some of the cons is most of these do carry management fee and carried interest, and the management fees can be a significant portion, of the total capital. So not all of your dollars are going into the actual investments. Not every micro VC fund is good at due diligence. Quality can vary widely. Most VC funds, particularly larger VC funds, remain pretty inaccessible to the typical individual investor. And the smaller funds, while more accessible, may not be able to participate at a meaningful level in larger later rounds, which kind of limits their ability to protect you as an investor in that company. They also tend to invest earlier. The smaller funds tend to invest earlier, which means there's usually a longer time for a first return. So incubators and accelerators, I think they're very useful for new founders who've never previously faced the many challenges of a typical startup. These are, they typically offer programs to support the growth and development of early stage companies. Some incubators and accelerators can also make startup investments. The top accelerators are highly selective and choose from a really large pool of applicant companies, provide a high number of investment opportunities, and typically provide more ongoing support than an angel network. Cons, for these larger accelerators like Y Combinator and Techstar, the investor participation is by invite only. Even though they've had some home run, their significant number of unicorns and home run successes, the overall success rate, based on the statistics I've seen, is pretty low. And there's typically little ongoing protection for the individual co-investors in the company. I also think that, some of the deal structures aren't particularly early stage investor friendly. So like the Y Combinator safe notes, I think are more company friendly than early stage friendly. I'm sorry, than investor friendly. So the last categories is venture studios, which is what VicTech is. That's a company that forms other companies and builds them from the ground up. Some, including VicTech, also providing the founding capital. There's a lot of pros with the venture studio model, and that's why you're seeing more and more venture studios out there. When we first started doing this, like I said, that term wasn't even out there, and there was really not people doing what we were doing, at least very closely, in terms of the business model. Some of the pros are that as a founding shareholder, in VicTech's case, we're the largest founding shareholder, we have a larger share of equity per dollar invested. And that translates directly to a higher return opportunity for the investors in the venture studio. So if you're investing in the venture studio and they're getting a much larger share of equity per dollar invested in their portfolio companies, that's higher upside for you as an investor. Good venture studios provide deep support to their portfolio companies, far beyond what a typical startup has available to it. And venture studios are also ongoing entities rather than, say, a one-time fund. So depending on when you invest in a venture studio, they may already have a portfolio of companies established at all stages of development. And if it includes later stage companies, that provides the investor's possibility for a more rapid return of capital. So at VIC, we have companies ranging from early R&D to sales growth stage, and someone investing in VicTech as part of the venture studio does have the opportunity for fast full return of capital. Good firms also have a large pool of technologies and a rigorous due diligence process to select the best IP foundations. And I think another significant advantage is because the teams are placed by the venture creation firm, there's fewer issues with problematic founders and team members who no longer serve a useful role. That's a fairly common problem with some startups, where the founders aren't the right people to grow the company, but they've already been involved and they put their efforts behind it. They've got their heart in it. It's hard to nudge them aside. In the venture studio model, the inventors of the technologies, typically professors at universities in VIC's case, are placed in technical advisory roles, and from day one they understand they're not carrying the football. VIC's carrying the football, and they're helping provide some coaching, regarding the technology. So those are the advantages. There's some cons too. First of all, many of these may not be open to the individual investors, and if open, the investment minimums may be significantly higher than say an angel investor network. And that's the case for VIC. For VIC investment into the venture studio, it's a $200K minimum. For investment into our portfolio companies through the VIC investor network, people can invest as little as $5K at a time into the portfolio companies. So it's a pretty big difference in terms of investment commitment. And again, just like all these other categories, their quality, the due diligence may vary. Many of these firms are regionally focused. Many are quite new and don't have well-established teams or deep experience in how you evaluate these technologies, and have limited resources for due diligence. So you always have to look at the specifics of the given company. I want to spend just a minute talking about what I was saying about the larger, for a venture studio, the larger ownership per dollar invested. That's a really important point. So if you look at the first row on this slide, this is comparing a venture studio model, and specifically VicTech's model, to a seed VC fund. So pre-investment ownership, basically the common founding shares in a company for a seed VC firm that's having companies approach it, already formed companies approach it for investment. They don't have any of those common shares. For VIC, we typically have 60% of the founders' ownership, with the other 40% going to our co-founders and management team. So then let's say we're taking a $500,000 startup investment into the given company. In the case of the seed VC firm, that's all coming from them. In the case of VIC's taking 20% of that founding investment, and the other $400,000 is coming from our affiliated VIC investor network. We always set the valuations very attractive. So we always, for a $500,000 investment, it's going to be a $750,000 pre-money valuation. If that's actually lower for the type of companies we've got, then we would seek if we were just going out on the open market. But for the purposes of this example, we'll just use $750,000 for both the seed VC fund and the VicTech. So with a $750,000 pre-money valuation and $500,000 investment, the seed VC fund gets 40% of the post-investment ownership, and that's all preferred. So they have 40% ownership, all in preferred stock. VIC's only putting in $100,000, and for that $100,000, we're getting 8% preferred ownership. But our 60% common ownership, is now diluted down to 36%. But in combination with the 8% preferred ownership, VIC has 44% of the total ownership for $100,000 invested, versus for the seed VC fund, 40% for a $500,000 investment. So if the companies do equally well, VIC would actually realize a 22x return unrealized at the Series A stage. But just looking at a typical Series A valuation, 22x unrealized return at the Series A stage versus a 4x for the seed VC fund. So that's one of the most important parts of the venture studio model. All these approaches I talked about have advantages and disadvantages, but let me open it up for questions now.
So we had some come through the chat box. I'll read them off here. The first is, how do companies lose from crowdfunding platforms?
How do companies do what from crowdfunding?
Lose from crowdfunding platforms. What's the downside to a crowdfunding platform?
For the company or for the investor?
For both.
So for the company, most companies going on these platforms are not successful raising capital. So they spend a lot of time on these platforms. Sometimes there's significant fees involved, and they're not successful raising capital. So you have to really you have to work to understand what your likelihood of success. Look at which companies have been successful, what stage, how do you really have confidence you can be successful? For the investor, the disadvantages are what I mentioned. The crowdfunding sites typically don't do deep dive due diligence on these companies they're putting on the crowdfunding side. They're earning their fees, and that's what that's what their business model is. They're putting these things out there at whatever terms the company wants to set them at, whether they're good terms or bad terms for the investor. So it's up to the investor to do the due diligence, and you have to sort through a huge number of companies to figure out which ones you like. So even though I've been on a board of a crowdfunding platform, it's of the approaches we described, that's my least favorite of the opportunities for individual investors to get involved.
Right. My experience is that the crowdfunding platform raises about 1% of your raise for you. The founder is on the hook for the other 99%. And I find that expectation is not always what startups think when they walk in. They think the crowdfunding platform is going to be the opposite. And so you have to think hard about these platforms because it's all on you for the most part.
Also, the crowdfunding platforms tend to over promote. So if a company's raised, let's say company A raises, $10 million, and $9.9 million that was from the founders raising from other sources. If they raise 100,000 on that platform and close around 10 million, the crowdfunding platform is going to say, hey, we helped that company raise $10 million, so they over promote in terms of their statistics on the sites. I've seen that multiple times.
Well, the other drawback I find is the crowdfunding platform doesn't always apply market rate valuations. I used to get deals from the crowdfunding platform where they had 50K of revenue, but they were worth $50 million in the tech space. And when they went to angels and VCs, it was dead on arrival because it was out of rate for market for sure.
Absolutely.
So what's your take on angel investing in the life science space? I've noticed that the angels really like short duration, short investment times, exiting at clinical 1 or clinical 2, not going all the way to FDA or to a product launch because of dilution. What's your take there?
Well, I think, as an angel investor in an early stage life science company, you better be prepared for a long wait. Exits can happen early if you can find an early buyer, but you don't know, and you're each step along the way, the company ownership, the board of directors at that company has to weigh the risk and return to a potential exit, early exit versus taking it further for a potential bigger exit, if they can even find an early exit. So while the goal may be an early exit, I think investors at the early stage, in the life science space, need to have a long time horizon. If they're investing across companies at all stages of development, they're getting some diversification, then their likely return time frame can be much less, because they've got some, and that's what we always encourage our investors in our network: put some of your capital in our later stage companies, put some in the middle stage, put some in the early stage with the highest upside. And that way you've got a diversified portfolio based both in terms of number of companies, but also across stage of development.
Well, I find for many life science deals, venture capital doesn't come in until clinical 1 or clinical 2. So you're pretty much having to use angels for the pre-NDA and pre-IND and those type of things. And so that seems to be part of the issue. When do you see VCs normally come in?
We've had them at all stages, preclinical and clinical. So we've had them even at the seed stage, but different VCs have different profiles, obviously. So some of the seed VC funds work pretty early, but their amount of capital they can invest is smaller. And then we also have strategic partner investors, like companies like Johnson & Johnson, that put capital in potentially at a pretty early stage if they're excited enough by the technology.
Great. Next few questions are about VicTech itself. Where is VicTech located, and is it a headquarters or is it distributed remote?
Yeah, so we have operations distributed all across the country. Headquarters are in Fayetteville, Arkansas, which is where I'm at. I'm an Arkansas native and returned here after grad school, but we have operations all across the country.
Great. And how many venture studios are you running at one time? Is it one venture studio with many groups, or is it multiple venture studios?
So we're one venture studio with a portfolio of companies that we're building.
Okay. And what's your source of deal flow?
So we have a team we call our opportunity assessment team. It's a 15-person team that meets every week, screening technologies available for license. These are typically from universities, and we've got good relationships with universities all across the country that are regularly sending us technologies to look at that they think might fit our model. And then we also just look at university websites and research papers and patents and dig for technologies as well. So we have a large number of technologies we review each year from a large number of sources, but the most common source is a university.
Okay, great. And what's the internal structure, the org chart of VIC? How is it organized?
So I'm CEO, and then we have managing directors in branch offices that can serve as interim CEO for new companies in their branch at the earliest stage. And then we have a full back office support that supports all the portfolio companies. So things like, from the first hire in a new company, they've got a complete benefits plan. We've got HR, accounting, scientific and engineering support that support all the branches. But each branch is run by a managing director who has equity incentives in the companies that are formed in their branch.
Okay. And then we'll come back to the mini masters question, but the next one is, tell us about your process with a startup. What do you do? Do you fill all the C-level slots, or do you do the development work or the lab work? What exactly is the value add of the venture studio or the process there?
So again, we don't work with existing companies that have been formed. Don't come to us and we put resources in. What we do is we take technologies out of universities and we form the companies. We put in place the initial CEO, and typically that's going to be one of our managing directors as an interim CEO. We'll hire the first technical product development person, to start doing the product development into the company with the seed capital that we put into the company, and then all the other initial work is the back office support and other stuff that I mentioned. As the company starts to mature, the VIC Investor Network might put a bridge round in, but by the time it gets to Series A, we're typically looking at recruiting a permanent CEO into the company. And by that time, the company is kind of tangibly exciting and not as far away from commercial realization. So you can attract the type of permanent CEO you want. So all of our managing directors are done it for people that have taken raw technologies all the way through commercialization and exit, but they're focused on the earliest stage. And then when the company gets a little further advanced, we recruit in a permanent CEO, and then the managing director moves on to their next interim CEO role.
So the next question is, who is at risk in one of your portfolio creations?
Who's at risk?
Who's taking the risk for the startup in that case?
So, obviously, VIC and the VIC Investor Network members are taking the risk. We're putting in the capital, and in the case of VIC, we're putting in a lot more than just capital. We're putting in a lot of resources, to support the development of the company. But we support the company all the way through the entire life cycle, and so it really provides, kind of an unmatched, compared to a typical startup level of support through bumps in the road, funding gaps, and other things, which is why we have a very low failure rate.
And then what about the individuals like the CEO and CTO? What level of risk are they taking? How much are they putting in?
So the interim CEO is a VIC employee, and they're not putting in capital into the new company. The permanent CEO that gets recruited may or may not be an investor, but certainly their compensation is heavily, as is the managing director, the interim CEO, heavily weighted on, to the upside. So they're taking the risk in the sense that they're working at below market salary in exchange for high level of equity compensation if the company is successful.
Right. And what percent of their wealth is dependent on that gig? In other words, what level of exposure does a permanent CEO have with one of these venture studio companies?
Again, some of the CEOs don't invest their own capital. They're all investing time and so on. But so that it varies a lot. But we never encourage anyone to put too much of their capital into a single entity. Just based on all these entities, even really good companies have risks. We don't ask them to put in. We're not. It's not like when I formed my first company, I put every dollar I had into it, and I made tons of mistakes, and I came very close to bankruptcy, although I eventually stumbled my way to success. But that we're not asking our team members to do that.
Okay, great. So are you raising funding now? Are you looking for limited partners at this point?
Yes, we are presently still raising capital. We think this is probably our last round that we'll ever take into VIC itself. And that's used both for our investments in our portfolio, follow-on investments, and expansion of our operations. But the reason we think it's the last round is just because of the exits we have coming up. We don't think we should have enough capital flowing back both to cover much larger number of investments into the portfolio and still distributing substantial funds to our investors and shareholders.
Okay, great. So what's the best way for people here to follow up with you afterwards to learn more about VIC and the opportunities there?
Sure. We've got a great portfolio of companies. If people want to look at individual deals or learn more about the VIC Investor Network, or if they want to learn about the investment opportunity in VIC Tech itself, just feel free to contact me at, you can use info@victech.com or my direct email, calvin.goforth@victech.com.
Great. I see it on the slide, but we're going to be moving to the next section. So during the following moments, if you could put that in the chat box, that'll help people email back to you afterwards. And then, of course, we'll make introductions with anybody that contacts 10 Capital. We'll make an introduction to Calvin as well. I want to thank you, Calvin, for a great presentation. And with that, I'd like to move to our first company presenter today, Jeff Missling of SpineTerra. Jeff, if you could go ahead and launch your slides, we'll check those out. While he does that, for those who came late, many of these companies are offering physician advisory shares. If you're a physician and you invest, you can also get advisory shares if you do additional work. If you're interested, we'll run a poll in a moment. Just register your interest to learn more about that, and we'll go from there. With that, let's go and pass it over to Jeff and go ahead and kick off.
Thank you very much, Paul, and pleasure everyone that's on the call today. Welcome to our presentation from SpineTerra. So happy to be talking with you today. So let me tell you a little bit about back pain in the United States. It is the most expensive condition we treat in the United States. $135 billion a year spent on back pain. That's more than we spend on cancer, more than we spend on heart disease. We do 5.8 million epidural steroid injections every year. It's just one component of that treatment for back pain. And the outcomes of those existing injections are not very good. Only about a third of the patients benefit from these existing epidural steroid injections. And this is what I'm going to tell you about today is SpineTerra's new drug for this application that greatly outperforms the existing treatment approaches with meaningfully better outcomes in this patient population. We have amazing support from some KOLs, really strong physician support from a recent market research survey, and a great proposition for healthcare insurers in managing their overall treatment costs in low back pain. Now, I want to tell you, if you're thinking about investing in SpineTerra, you're investing more than just our drug for back pain, but you're investing in a platform technology. So we have created this technology we call a micro suspension that enables a sustained release, meaning a long, slow, steady release of the active ingredient over 90 days from a highly concentrated formulation that can be injected through small needles and catheters into tight anatomical locations like the lumbar spine. We see multiple follow-on opportunities for this technology, for example, regional anesthesia and postoperative pain, addiction treatment products for opioid use disorder. But as the name of the company would suggest, at this stage of development, we are highly focused in this large unmet need in back pain. So let's dive into the back pain patient journey. If you know anyone who's experienced back pain, this is probably what they went through. They start out with oral medications, physical therapy, maybe just told to wait, right? Just take a conservative approach. But if that pain persists for 4 to 6 weeks and it's not improving, they'll often get an MRI. If that MRI shows a herniated disc in their lumbar spine, then they're a great candidate for an epidural steroid injection, which again is just a cortisone shot into the epidural fat adjacent to the nerve root that is causing their back pain. And we're doing this 10 million times a year in the United States. If that doesn't work, then the patients are moving on to really expensive surgeries, which have not that good of outcomes, or spinal cord stimulation implants, which again are really expensive interventions. So zeroing in then on these epidural steroid injections in the middle of the care continuum, there are 4 drugs available today that are used for this purpose. If you look at the data that's been published, and there are hundreds of papers that have been published on the efficacy of epidural steroid injections, only about a third of the patients receive adequate pain relief from a first injection with any of these drugs that are out there today. And that efficacy is short term. Even if it does work, about 2/3 of the patients will go on to get a second injection within the first 60 days of treatment. So the outcomes are poor and they're short term. This is the problem that we set out to address with our technology. So the drug we've developed, we call SX600. We designed and developed this formulation to achieve a long-term localized delivery of the key active ingredient, which is dexamethasone. Dexamethasone is the steroid that doctors prefer for this application today. It's used in over 50% of the cases. What we have done is encapsulated dexamethasone into these tiny biodegradable microspheres that provide slow, steady exposure of the active ingredient right at that nerve root, which is the source of the patient's back pain. So what do I mean by that? Well, here's some data showing the exposure profile from a single injection of our drug, which is the green line on this graph, compared to an injection of the drug that doctors are using today. So you can see we give about a 2.5-fold higher dose, but we minimize this peak exposure systemically, which is good to avoid side effects systemically, but we extend the duration of exposure out from 1 to 2 days all the way out to 90 days. And we have confirmed this exposure profile in a human clinical trial that we completed just last year. So this is a truly different product, right, that would be available to patients after this is FDA approved, a totally different exposure profile. And with the clinical data I'm going to share with you, it did translate into much improved outcomes. So again, diving into this clinical trial, we called it the Salient study. There were 3 arms in the study: a saline placebo group, a low dose group of our product, 12.5 milligrams, or a high dose 25 milligram group. These patients received a single epidural injection. Our product doesn't change anything about the procedure that the doctors are doing 10 million times a year. So they just used our drug in their clinical practice as part of this study. And we followed the patients out for 180 days. Patients had an app on their smartphone and they reported their worst pain score of the day every evening during the study. So we had really reliable and consistent pain reporting by the patients in real time over the life of the study, which is so important in a pain trial. Here's the key data. So our primary outcome measure was the proportion of patients that achieved a 50% or greater reduction from their baseline pain score. So that is highly clinically relevant, right? I mean, if you came in with a score of 8 out of 10, self-reported, to meet this threshold, you had to be a 4 out of 10 or lower. You can see in our high dose group, 71% of patients met that threshold compared to only 13% in the placebo group. We had a nice dose response, and that response was maintained in 64% of the patients through 180 days. So this data has doctors really excited. In fact, right before this call, I just was on a Zoom call with a KOL from Toronto, shared him the data. He got all jazzed about it. He was like, Jeff, how can I get involved? The doctors who have seen this data, who are treating these patients, are really excited about the potential of this product. Here's a couple KOL quotes from the US. I'll just point to the bottom one here. Yesh Navalgun, who's the chief medical officer of one of the largest ASCs on the East Coast. SX600 fits perfectly into the needs of the patient, the doctor, and the payer. Now, these KOL opinions align very well with some market research that we just conducted. We surveyed 63 physicians. A third party did this, right, on our behalf, presented a hypothetical product profile based on our clinical data, and asked these doctors, how likely are you to use this drug in your practice? And they reported 70% adoption of our drug, which is extremely high for a market research study of a new drug in development. And in fact, 40% of the doctors said they would convert 100% of their patients to this drug if it was available. Now, that level of adoption translates into a huge potential market for our product. We're doing 5.8 million lumbar epidural injections every year in the United States, right? 10 million in total, 5.8 are in the lumbar spine. 3.5 million of those are not repeat injections, right? So it's common people are getting repeats. 3.5 are just first injections. So we think that's our addressable market. At $600 per dose, that would translate into a $2 billion market. And again, based on this market research that we just completed, 70% adoption implies peak sales potential of our drug of $1.5 billion. So a huge potential market here for the drug in low back pain. We have some competitors in the space, and I'm just going to tell you, we're the only company that has demonstrated a significant advantage over the current standard of care. None of the other companies have come out with clinical data showing anything that's differentiated from these generic drugs that are being used off-label today. So quickly, a little bit about the team. We've raised about, $30 million in the history of the company, $7 million in DoD and NIH grants. Back pain is a huge problem for the military. Just under 11 million in angel investors, and our angel investors include former or retired executives from many well-known companies: Allergan, Medtronic, Mallinckrodt, Stryker. And earlier on in the company, we had a strategic partnership with an opioid manufacturer who went bankrupt due to the opioid lawsuits, but that did provide us $12 million in funding. So very strong support from the DoD and individual angel investors to get to this point. We have great intellectual property, 5 issued US patents, and our 6th patent was just allowed. So very well protected IP in this space. Our leadership team, I want to highlight our Chief Medical Officer, Lee Techner. He's really driving the next stages of drug development for us. 22 years of clinical research under his belt after 15 years of clinical practice. Years of experience developing drugs for pain indications. Our board of directors, I won't run through all the people here, but just look at some of the companies they're coming from: Seidman Therapeutics, Elvigen, CNS, Genentech, Medtronic, and Shaheen Lakan, who trained at Mass General and Cleveland Clinic, and who's now the CMO at a digital health company, Click Therapeutics. So we've attracted some really great advisors to the company from the Mayo Clinic, and one of the top pain doctors in the country, Tim Deere, on our advisory board. So just a great group of executive advisors surrounding our team here. Now, what's our ask today? We're out raising via a convertible note, a $5 million round. The terms of the notes include 10% interest until the point of conversion. They're going to convert at the lower of a 20% discount to our next equity round, or at a $24 million valuation cap. So that's actually a great value for a company that's sitting on positive phase 2 clinical trial data, and the notes come with a 4-year warrant. What are we going to do with this money? We're going to execute the next stage of development that's critical to advance our drug with the FDA, which is a mini pig epidural toxicology study. We've already done toxicology studies in rats and dogs. We've shared that data with the FDA. They looked at our rat data and said, we really don't think rats are the right model for this type of drug. Even though we told you at an earlier meeting that it would be fine, they told us after they saw our data, they wanted to see a second non-rodent species. That is the critical next step for our drug development, and that's what we're seeking the funding to do. It's a very low risk study. Again, we've already done this study in dogs and had no issues, and mini pigs are about the same size as a dog. We've actually done a 14-day pilot study in miniature pigs that had no issues. So again, this is really a regulatory activity to move drug development forward. We firmly believe, based on the discussions that we're already in, that with this mini pig tox study complete, we're in a great position to strike a strategic partnership with a fully integrated pharmaceutical company that could complete the late stage development and FDA approval of our drug, which is our primary exit strategy. I mean, that's what we want to do. We have taken this drug far enough. We get this mini pig tox study done, satisfy the FDA, and companies like some of the names that I've put on the slide are going to come to the table with some money. And as I mentioned, we're in discussions with many of these companies already. They're following us, they're watching us, and they're excited about the potential of the drug. Paul, I think I've saved 3 minutes for questions.
Great. So the first question, if you go to slide 9, is in the number of patients.
Yeah, it was 56 patients in total in the clinical study, split fairly evenly across the treatment arms. Sorry, I went a little bit past it there. So it's 56 in total.
Okay, great. And then on slide 10, how many patients are in that bar labeled 71%?
Again, that's the 25 milligram group, where there were 17 randomized into the 25 milligram group. So 17 in that group.
Okay. Next question is, do the patents cover just the delivery method, or do they cover any new drugs?
They are very broad. They cover a whole range of active pharmaceutical ingredients. So the primary claim would read a pharmaceutical composition comprising. It describes a class of biodegradable polymers. broad classes of active ingredients. And then really what it's about is the narrow size range that we're making the microspheres in, which is different than anyone else who's using this type of technology. That's really the scope of the patent. So we can absolutely formulate other active ingredients into the system for both a technical and an intellectual property standpoint.
Great. And what's the time frame to FDA approval? Where are you on the path, and how far to the approval do you think that's going to be time-wise?
I'll go back to that clinical development plan slide here. So again, we need to complete this epidural tox study in miniature pigs to satisfy the FDA. Then we need to complete late stage clinical development. And so that would involve a significantly larger fundraise, or preferably a strategic partnership. Right, once you're into late stage clinical trials, you're starting to build a commercial team, you're starting to prepare for launch. That really is more logically done by a fully integrated pharmaceutical company than a small biotech like we are. So we think we're about late 27 when we complete clinical development and be ready to submit an NDA. But again, that would be somewhat dependent on a strategic partner who takes over the development of the drug.
Great. Last question is, what were the rejection rates in the preclinical studies?
In the clinical studies?
Or preclinical, whatever you've done so far.
I'm not sure if I understand the question, but the non-clinical studies we've done, which are extensive, are highly supportive of further drug development, now corroborated by the safety data from our 56 patient human clinical study.
Okay, great. I don't think you've done the clinical studies yet, so I was wondering if you had anything else, but you answered the question there. Okay.
And whoever asked that, if I didn't get at what you were asking, please follow up with me outside of the call, and I'd be happy to dive in and make sure that the question is addressed.
Right. Well, when you get back, you can look at the chat box and you can type in some replies there if you have further information, if that helps. And with that, we'll go ahead and move to our next presenter. Thank you, Jeff. And with that, we'll go on to Anita Davidson of Creative Biotherapeutics. Anita, if you want to launch your slides, we'll kick off and reset the timer.
There. Can you see my slides?
We're ready to go.
We are ready to go. Thank you, Hall, and I'm happy to be here. So Creative Biotherapeutics is an emerging biotech that starts research where other therapies fail. The problem we are solving is that drug-resistant cancers are incurable, which impacts 600,000 patients each year in the US. These are the top 10 cancers in the US last year alone, and these are the number of recurrences in each of these cancers. CBT is focusing on the $21 billion market of these 6 cancers, with looking for a beachhead target market of metastatic breast cancer. So our solution to the problem is CBT 300, a biologic that reverses drug resistance. So when tumors develop, they place a protein on the surface called GRP78. That protein causes drug resistance and blocks immune cells. CBT removes that GRP78, allowing the immune cells to get to work and restoring the cells to more normal. So does it work? So to show that CBT 300 is effective, we ran a head-to-head comparison with standard of care Keytruda in 4 different mice tumor models. So for example, we've got melanoma in the bottom right corner. It had a 63% increase in survival compared to 0% with Keytruda. Likewise, with lung cancer, CBT 300 demonstrated a 75% increase in survival compared to 37% with Keytruda. Now, this was a standalone test with CBT 300 without any combination. So what we also did is we did a combination with this breast cancer study. So this is drug resistance. Each of these cancers are drug resistant. So we did a breast cancer study with a maximum dose of doxorubicin on the bottom and a combination of CBT 300 and 50% of doxorubicin and in the middle. And you can see there's a significant reduction in tumor size at the end of the study. So the last studies were in mouse disease, but these studies are with patient-derived tumors grown as spheroids. And current studies are showing that use of tumor spheroids is highly predictive of efficacy in clinical studies. So these are 3 highly drug-resistant tumors that we tested with CBT-300 and in combination with doxorubicin. As you can see from the pictures, alone or in combination with low levels of dox, there is significant tumor regression. So imagine this scenario. Carolyn follows up with her oncologist. Your breast cancer has returned. You have suffered through 2 rounds of chemotherapy. Immunotherapy with Keytruda only targeted 20% of your cancer, and ADCs don't work for you. Your cancer is resistant to all other therapies. Fortunately, there's a new biologic available called CBT-300. It isn't toxic, has lower costs, and it works well with smaller doses of chemotherapy that could increase your survival for years. What do you think? If Carolyn had that choice, it would have been yes, please. Unfortunately, she didn't and died at the age of 49, leaving 4 young children, a husband, and a thriving music career behind. And I know this because Carolyn was my college roommate. But we hear these stories every week. So CBT-300 can be a standalone or in combination to help change this scenario. And with these current therapies, we can do combination, or we could be a standalone. And so CBT-300 will check off all the boxes. CBT has strong intellectual property protection. We have 5 therapies that are protected under our composition of matter patent. We've had excellent feedback from key opinion leaders, and we are ready to complete the drug scale-up and the toxicity studies that we need to go to the FDA for clinical trials. The CBT team has the background, the track record, and the vision to succeed. We've had 4 successful startups with 3 exits, 40 publications and patents, and over 100 years in healthcare and pharma experience. We have an efficient business model with a strong advisory board, top-tier CROs, and excellent support companies. We are currently vetting potential board of director candidates, including key opinion leaders and investors. As we move toward clinical trials, we will scale up our staffing internally using the commercialization experience of Syneos Health as a bridge for our market entry. As the beachhead target market, metastatic breast cancer has a SAM of over 43,000 women every year, and that's based on the annual US death rates, and that has been consistent for the past 30 years as a result of drug resistance. So regardless of the advances made, that there's still over 40 to 43,000 women that die each year. We are moving CBT-300 to the market in Q2 2026 as a single monthly injection of $5,000 for metastatic breast cancer with a projected gross revenue of $130 million year 1, building on market expansion in breast and other cancers. In addition to MBC, metastatic breast cancer, we have strong data to move into these other drug-resistant cancer markets based on our preclinical data. We have raised $2.2 million to date in diluted and non-dilutive funding. Our current Series A raise of $3 million is for drug scale-up, toxicity studies, and regulatory fees. We are offering a participating convertible preferred equity plan for 15% of the company at a $20 million valuation per Keiretsu Mid-Atlantic. This includes warrants at 50% for the first $1 million raise, 30% for the second $1 million, and 10% for the third $1 million in this raise. We are considering a wide range of exit possibilities for CBT-300 or the other 4 compounds that we have under patent. So with considering individual disease markets for partnering, we can branch off the cancer markets or some of the other markets that we have potentially to develop. So we have a lot of possibility with working with large pharma, and with our CEO's experience with Abbott and AbbVie, we have a high level of confidence with regards to what potentially could could develop as far as a partnership. Since our patents cover multiple products for multiple potential diseases, we do need to be mindful of the exit strategy so that we are breaking off pieces as opposed to the entire company. So please consider working with us as we change the paradigm for how drug-resistant cancers are treated for the 600,000 patients that suffer from drug resistance each year. And at this time, I'd like to invite you to link into my Calendly if you would like to have a follow-up. And I am open to your questions and comments.
There was a question posed, is all MBC drug resistant? And Donald gave a response to that, but would you like to add on to that?
Actually, I would like to invite our CEO, Dr. Donald Davidson. He's all and to help with the question and answers, so that he is the creator of the drug and the scientific expert.
Great, Donald.
Thank you. Yeah, so I tried to answer the best I could. Not all metastatic breast cancer is drug resistant at the start, but usually with chemo or with Keytruda or any of the targeted therapies, about 80% becomes resistant over time.
Great. So you have many opportunities to apply this technology. Why are we going after MBC first?
Well, we selected MBC first because of the ongoing issue year over year for 30 years. I mean, as you can tell, there is an emotional connection with my prior roommate, but we also, I have heard and talked to people over the last several years, and it just persists, and the devastation to the families continues. And because that market there is nothing for that population, and our drug targets from the end where there isn't anything for them, we wanted to start at that point and then move backward and expand from there. So from a market standpoint, it has a high predictability of revenue generation, but also a high impact with regards to patient care.
Right. Is the DPT equivalent to an MD?
No, it is not. So it's a doctorate in physical therapy. So I've been a licensed physical therapist for nearly 40 years. But it is heavy on research and such. But I've been a CEO for the last 25 years in one of our other businesses.
Who are the likely acquirers if you exit at clinical 1 or 2?
We would be looking at companies that, for instance, at AbbVie, we would be looking at Genentech, we would be looking at potentially probably not Merck, but we could potentially market it to Merck because we can make Keytruda better. And so we can look for those to collaborate with those companies.
What's the time frame between here and clinical 1 and clinical 2? What's the number of years or months is that case?
Okay, so we're aggressively targeting for Q2 of 2026, but that of course, depends on getting this funding. So it's going to take us about 5 months to do the drug scale up, another 6 months for the rat toxicity. So we anticipate next summer being an IND, and by Q3, Q4, preparing and being in phase 1 clinical study, which will be a 3-month study with patients that actually have cancer, so that we can look at efficacy as well as the toxicity. And then we will follow with a 12-month phase 2 clinical trial. One of the things that is key with regards to oncology is since 2015, 70% of the new oncology drugs have been approved by the FDA after phase 2. And so we can continue additional trials, but they have approved those. And we anticipate with our lack of toxicity that we aren't going to have any problems.
So 30% of drug candidates fail due to liver toxicity. Do you see that being an issue here? You're saying it's less toxic. Can you tell us more?
Yes, I'll be glad to take that one. So small molecules do have liver toxicity. This is a biologic. Biologics get degraded like proteins. So it's not a big problem. And we have not seen any liver toxicity in any of our mouse studies yet. So usually antibodies don't have any problems with liver toxicity. So this is similar to that type of a molecule, and we don't expect it.
And what's the current status of the patents that you have? How many have been filed and how many have been awarded?
So we have 5 different compounds under our composition of matter. So those were awarded in February of 2021. We also have use patents worldwide and domestically that are in process that we should have final determination for the use in virus with COVID, because it also has breadth beyond cancer for this therapy. So we have also developed and have started to apply for a next generation product too. That so we're continuing to add more intellectual property to the portfolio, but we are covered until 2041. Pretty much we have a kitchen sink patent according to the patent lawyers. So we've got quite a bit of coverage.
Have you filed for a freedom to operate?
Yes, we haven't had any issues with legal review. And we've also, as part of our National Science Foundation grants, that has been an issue that we've been vetted very heavily and very, very carefully with that. So we're very confident that we don't have any limitations there.
Great. Are there any other questions from the audience? A mouse without liver toxicity. Okay. I think they're understanding what we're talking about here. That sounds good. Any other questions at this point? I think that takes us to the end of this section. Thanks so much, Anita and Donald. And with that,
Thank
let's
you.
go ahead and bring up our next presenter. We have with us Glenn Markov of Rejuvenation Therapeutics, so Rejuvenation Technologies. So Glenn, if you go ahead and launch your slides and kick off.
All right. So I am Glenn Markov, the co-founder and COO of Rejuvenation Technologies, a company dedicated to addressing aging at the mechanism of its sort of fundamental biology and reversing it to treat age-related disease. We got started in 2018 out of a discovery from Stanford University and a lab that I did my PhD in along with John Ramirez, the co-inventor of the technology. John was doing his PhD in neuroscience at the time and was studying the, amongst other things, the effects of telomere length on human cells and looking for ways to extend telomeres. I was doing my PhD in genetics on epigenetic reprogramming. I'm looking to turn back the clock, if you will, in epigenetic sense, turning cells into stem cells. The focus of our company is on the telomeres. So telomeres are an aging clock. They are a bit like the plastic tips at the end of shoelaces, and they shorten with age. They shorten every time cells divide. And when they become too short, the cells start to break down. So you have a loss of cell function. Cells can trigger this chronic DNA damage, which is a hallmark of aging. And you can see on the figure on the right, you have a number of hallmarks of aging that are all underlying short telomeres. So telomeres are very interesting. They have a wide range of distributions, even between chromosomes in a single cell. They are effectively a repetitive sequence of DNA, and they form a loop at the end when they're sufficiently long. And that solves the one of the core issues of having linear chromosomes, which we do. Of course, all mammals have linear chromosomes, and in fact, the rate at which telomeres shorten are a very good predictor of lifespan in mammals. And the idea is, and this has been shown in mice, that if you can extend telomeres, you can extend lifespan, delay the onset of age-related disease. What happens is when telomeres become too short, cells begin to function poorly. Our natural regenerative capacity that we have as young adults begins to break down, and age-related disease sets in. These types of chronic diseases have had very limited treatment options because the fundamental mechanism of aging is not being addressed. Put into another way, telomere extension enables regeneration because it allows cells to divide and repair the surrounding tissues. The goal of our company and the technology that we have is to treat patients who are shown in this sort of orange circle here that have very short telomeres and bring them into the Goldilocks zone, where telomeres are about 10,000 bases in young adulthood down to about 5,000 bases before you see the onset of classical signs of aging and age-related disease. This happens in all of the tissues of the body, but in some tissues it happens faster than others, and there's where you see the onset of specific diseases. The solution we have is a synthetic messenger RNA encoding for telomerase, the enzyme that naturally extends telomeres in the body. The effects of a single dose of telomerase messenger RNA in human cells is profound. It reverses the equivalent of a decade of telomere shortening. With 3 doses, we reverse about 20 years of telomere shortening. Now you can see the data that's published in the bottom left. Here we see that the blue graph represents the telomere length of cells treated with telomerase messenger RNA. And you can see how the distribution is shifted compared to the orange, which is the distribution of cells that are not treated with telomerase messenger RNA and approaching the end of their lifespan. When we do this treatment, not only do we see an extension in telomeres, but we see a reduction in senescent cells and a reduction in enlarged cells. This allows the cells to continue to divide a finite number of times before, again, they reach the end of their lifespan. And we can treat them again to, again, reverse that aging clock. And as a way, this provides a very good approach as a drug to keep, to treat age-related disease and to maintain kind of that youthful phenotype from perspective of telomere length. The messenger RNA that we deliver, it codes for telomerase, which is the natural enzyme that extends telomeres. It has been designed and honed by evolution over billions of years to do just that. And as a result, it does a job very well. We have very excellent temporal control because the nature of the messenger RNA results in transient bursts of the protein, which extends telomeres and then is degraded. And we can go in and do multiple doses as necessary. We deliver this with lipid nanoparticles to different tissues in the body to treat specific diseases. This is really the best-in-class approach that we have compared to other alternatives to extend telomeres. There are indirect activators of telomerase. The most direct ones that work well from a telomere extension perspective involve delivering the enzyme as DNA. However, these are not very safe because when DNA is entered into a cell, in some fraction of the cells, that DNA is now integrated into the genome, and that results in long-term expression. And this has been shown in both animal and human studies. As well, delivered DNA is often done in a viral approach, which is much more immunogenic than a lipid nanoparticle approach. There's also been many efforts to develop small molecules to activate telomerase, but it's highly regulated in the cell. And so these have not been very effective. Telomerase has been studied for years in the lab as it relates to cancer. It's one of the prerequisites. When telomerase is on permanently, it allows cells to divide indefinitely and confers immortality. Very useful cell, very useful tool in the lab to have. As a therapeutic approach, the way to get around this problem is to deliver telomerase transiently, and that's accomplished with our telomerase messenger RNA. This is the way cells in our body, such as T cells or stem cells do. They activate telomerase naturally throughout life to maintain telomere length. And our feedback from the FDA so far has been positive in this regard. We've shown that our telomerase activity is very transient. There were no concerns about the telomerase approach. And we have not seen tumor incidence in a number of our animal models. The diseases that we can treat are numerous because it spans the landscape of age-related disease. And as such, the approach is really a platform approach. We're limited largely by bandwidth and the delivery technologies, which are evolving every day. Our approach has been to focus on diseases that show the strongest clinical risk-benefit ratio and the strongest rationale scientifically, as well as places we can deliver to. And so that really narrowed our list down to the lung, liver, and the bone marrow or immune system. In patients that have mutations in telomerase, they experience a number of symptoms and premature aging. But what we see in those patients is a preponderance and ultimately death due to pulmonary fibrosis, liver cirrhosis, or bone marrow failure, or a combination of those. We have multiple projects in development. This includes the pulmonary program, which can target the lung and can be used to treat not just pulmonary disease, but also, or not just pulmonary fibrosis, but also potentially COPD and fibrosis from long COVID. Our liver program, we're focusing on liver cirrhosis and alcoholic hepatitis, but this can also be used to treat earlier stage liver disease like NASH, as well as cholestatic liver disease, all of which show telomere shortening, and not just telomere shortening, but also a more rapid progression of the disease that is telomere dependent. A little bit of data about our deliver technology. For the lung, we had to invent our own lipid nanoparticle in order to target the lung safely and effectively. We used a combination of lipids, including one novel proprietary ionizable lipid and several off-patent lipid components. These we mix in a microfluidic manner or an impingement jet mixer to generate lipid nanoparticles that are around 50 nanometers in size, and we deliver this intravenously. We see very good lung tropism, as shown in this slide here, where we're delivering messenger RNA, in this case encoding luciferase, the firefly enzyme that generates light, which we can detect, and which tells us the biodistribution. You can see it's largely lung. We've repeated this now in both rabbit and non-human primate models to confirm the lung tropism. Within the lung, we've looked at the cells that we're hitting, and we are able to deliver to the vast majority of the alveolar cells, including the alveolar epithelial cells, which is our target cell type for the pulmonary fibrosis indication. So this here graph is showing a dose response of dose compared to number of transfected cells. We've done, since our founding in 2018, we've done many animal studies demonstrating the fact that our drug can treat a number of the disease equivalents, the human disease equivalents in animals, including showing a large reduction in pulmonary fibrosis, increase in lung function using forced vital capacity, which is the endpoint the FDA uses for clinical approval, as well as improvement in a number of other features such as lung architecture. We've also done a similar panel of studies in our liver program, seeing that we get a large reduction in liver fibrosis, as well as improvement in a number of other parameters of liver function, such as reduction in DNA damage and reduction in high-grade inflammation. We have a very good safety profile, as judged by several parameters, including rapid drug clearance. Here I'm showing the graph of drug clearance in plasma, looking at both the lipid and the messenger RNA in a 24-hour window. You can see the vast majority of the product is gone. We see slower clearance from the lung. In this case, this is the lung LNP, as you would expect. We've done a histology panel that has been reviewed by board-certified pathologists, examining in microscopic detail a number of different tissues and seen no moderate or severe findings. We've done a clinical chemistry panel. We've looked at immunogenicity after multiple doses, including high and low dose. And I looked at a number of cytokines, some of which are shown here, to make sure they go back to baseline. We have multiple strategic paths that we're considering as we move forward in our program. We are looking at both the pulmonary program, and the milestones are listed here, which include manufacturing, scale up, and GLP, which we've started, and GLP toxicology in rats and non-human primates. And then a phase 1 clinical trial in pulmonary fibrosis patients, where we'll be looking at biomarkers of telomerase activity, disease progression as well as safety to decide on the dose to be used and dose frequency to be used in the phase 2 trial. Our hepatic program is following a similar trajectory. We're looking to do a pilot non-human primate study there to find the target dose, which we've done already in our pulmonary program, and then a similar set of endpoints. The possible exits include both looking at comparables from a public market perspective, as many biotechs go public after their clinical data readout. And so here I'm showing market caps of 2 companies that have assets in development for pulmonary fibrosis. But in both cases, these are molecules that are largely kind of early generation, focusing on the fibrotic aspect. We also have 2 examples down here of assets being purchased by large pharma partners that we've also engaged. A summary of this financing round is shown here. We're raising a $2 million round. This is on the back of having raised already nearly $9 million from our previous investments. We have about $400K remaining in the round. We also have 2 grants starting this month. End of the week, we're starting our California Institute of Regenerative Medicine grant for our pulmonary program, a $4 million grant, as well as a $2.6 million grant focused on liver disease from the National Institute of Health. Our use of funds will include scale-up manufacturing and the toxicology studies, as well as patent prosecution of our number of patent families, which importantly is not covered by our incoming grant funds. A summary of our patent situation is here, where you can see that we have our core IP around a telomere extension technology and telomerase messenger RNA, which we have an exclusive license for and granted in multiple countries, all the major jurisdictions, as well as 2 public patent families, one for each of our main lead programs and a number of provisionals that we're not showing here. We have a team with deep expertise in the pharmaceutical industry and the RNA lipid nanoparticle field. The management team is John and myself. We've brought on board Susanna Tulach, who's as a director of our scientific research team, who's been a part of the $3 billion GMP production environment and is a professional project manager. We also have folks with deep expertise in both lipid nanoparticle development and execution, such as Viktor Napov, who developed an LNP that was then sold to BMS for $100 million. We also have folks like Craig Shimasaki, who founded 9 biotech companies, taken 5 public, Joy Cavagnero, who is advisor for Pfizer on their COVID vaccine, Patty, who developed the messenger RNA and the manufacturing procedures for Arcturus, a public NASDAQ traded company, Helen Blau, our graduate advisor and National Academy member, Stanford professor, as well as Doug Kornbrust, who has experience on over 100 RNA drugs in the preclinical space. And a clinical advisory board for our pulmonary program is shown here. So a summary, we have a great investment team, including Y Combinator, who was our first investor, who put in $1 million, Coastal Ventures that put in $2 million on the back of the YC investment, number of funds and angel investors. Thank you.
Great. We have time for one question. What does reverse aging clock mean?
It's the aging clock is the telomere clock, and telomeres are sort of kind of like the tips of our shoelaces. They shorten with age, and we're able to re-extend the telomeres. And so we're able to allow the cells to divide more. And you can think of the aging clock being rewound, about 10 years with a single dose of the drug.
Great. Well, we're at the end of our time. I think you covered all the major topics there. We did ask the audience for their interest on the poll. And with that, we'll go and close out our session today for that. Thank you, Glenn. Thank you, Anita. Thank you, Donald, for your presentations there. And with that, again, if you are a physician and have interest in advising these companies, please contact us and we'll be glad to connect you with the right groups, as we have these and other life science companies that are looking for your support. With that, we'll be back for another round of events next week, and looking forward to seeing more of you guys come in with more questions. Thanks so much for asking great questions today. And with that, we're going to wrap it up for now.